Apresentaremos agora a palestra O Cérebro Alfabetizado, como aprender a ler, depende e muda a organização cerebral da linguagem falada. Eu convido à mesa o moderador, professor Augusto Butchweitz, e convido também o palestrante Kenneth Pugh, da Haskins Laboratórios. Eu passo a palavra ao nosso moderador, desejando a todas e todos um ótimo trabalho.
Bom dia a todos. Muito brevemente, eu gostaria aqui de agradecer a este convite, a esta oportunidade, e também especialmente ao Dr. Kenneth Pugh por estar aqui. Ele é nosso parceiro já há muito tempo, e nós convidamos ele em cima da hora e ele veio. Então, eu acho que ele tem para trazer para vocês evidências bastante interessantes sobre o processo de alfabetização e aprendizagem da leitura e as alterações e adaptações do cérebro humano. Dr. Pugh, ele é presidente do Haskins Laboratórios, que é uma instituição centenária já, que estuda a fala e a leitura, e também professor da Universidade de Yale e da Universidade de Connecticut. E se eu fosse ler o currículo dele, a gente não ia parar nunca. Então, por favor, Dr. Kenneth Pugh, you have the floor.
Thank you. Can everybody hear me? Okay. So I will stand over here, if that's okay. First of all, I want to begin by thanking Hinan and all the organizers. It's a great honor to be here with you today, and I look forward to sharing our work and some work that will connect with the following speakers, with Augusto and his team. We are collaborating on some of our projects. Now, I'm here to talk about the neurobiology of reading and language. What can the brain tell us about learning to read? I come from Haskins Laboratories, where I'm the president and director of research. We are officially or formally part of both Yale University and the University of Connecticut. That's why I have a joint faculty appointment both at Yale and at the University of Connecticut. And we have been around for many years, 87 years. The lab is a place where we focus on the biology of human communication. So we are looking at how people communicate in every way, shape, and form, including sign language and oral language and written language, reading and writing. And so my presentation today, which will go into lots of terminology about the brain, for those of you who are not in neuroscience, don't worry about all the details, all the names of these different brain regions. My job, if I do it correctly, is to convey to you what neuroscience can tell us, what the promise is, and also where we need to be cautious, where sometimes people make claims that are too far beyond the data. So for educators, for researchers in other fields, it's important to understand what the brain research is telling us, but also what it's not telling us, what it's good for and what it's not good for, and where it can impact our understanding of reading. I will conclude my talk today with a very optimistic view of brain research as a vehicle to provide what we call exquisite sensitivity to individual differences. It's a wonderful lens on understanding individual differences from one child to another. And the more deeply we understand these biologic differences, the more we can tailor the learning to the brain. This is the hope and dream of educational neuroscience, but at present we need to be very careful because sometimes people claim more than the science supports. And at Haskins Laboratories, we are very committed to understanding language in all its form, as I said. And what that means is we pay a very special attention to how written language, reading and writing, depends upon and sits on oral language, the biologic specialization for talking and listening. So a lot of the data I'm going to present in the first half of my talk today will be geared toward how learning to read depends on speech, and actually how it changes speech. And my dear friend Ana Luisa Navas, who studied with us did her graduate work at Haskins when we were many years ago, when we were younger, has also alluded in her talk to the ways in which oral language and written language depend upon one another. And so we do work on a number of topics that relate to how language and reading depend upon one another. Historically, we do a lot of work on how the mechanisms of talking and listening shape reading and writing, and how they're changed by reading and writing. And you'll see lots of data from me as I run through this on those relations. We also are known as the place where the orthographic depth hypothesis was created. My colleagues Ram Frost and Len Cates, in particular, did work to codify each written language in terms of the transparency. You've heard a lot about it in the last 2 days. Some languages, like Portuguese or especially Finnish, are very regular with regard to the orthographic phonologic relation. English, not so much. Unpointed Hebrew, even less. Mandarin Chinese, with its phonetic and semantic radicals, even less. But you'll see data from me on how, at the brain level, some of these differences don't matter so much. How we see a great deal of universality with regard to how written language depends upon oral language. So here's the question for my talk, and I suspect for some of the other talks coming afterwards from Augusto and his colleagues. Can a better understanding of neurodevelopment, and this means gene, brain, cognitive relations, can that actually improve the way we teach or remediate reading, especially because as we understand the brain better, we can see what is easier or harder for the brain at different ages. Some children learn to read younger. Some children are not introduced to literacy until much later. The brain is maturing and changing. Can an understanding of that provide insight into how to tailor the learning? Also, different abilities. I do a lot of work, if those of you are familiar with my work, you know that I do a lot of work on dyslexia and reading disability, and I'll talk about that today. So on top of poverty and limited access to resources that can make literacy difficult for so many kids, for significant numbers of kids in all languages, there will be further difficulties that are congenital, that are brain-based. We call this developmental dyslexia. And languages, of course. Is learning to read, here's the question that we've heard many times in the last few days. Is the way in which the brain builds the reading circuit, the connections, similar or dissimilar in a transparent language, relatively transparent language like Brazilian Portuguese, relative to an opaque or a deeper orthography such as English? So let's begin at the beginning. At Haskins Laboratories, where I work, if I don't have a slide like this, even though I'm the president, they will fire me. Because we really care about how written language and speech relate to one another. And so I'll start with a couple of overview ideas. Speech is a biologic specialization. What does that mean? We have specialized brain systems that are wired to talk and listen. When a baby comes into the world, the baby doesn't need to go to school to perceive speech. I mean, to put it in a silly way. The baby needs to be talked to a lot, needs to be given great stimulation in those first years. But the brain carries with it a lot of biologic machinery that has evolved over a very long period in our species that makes us wired for speaking and listening. On the other hand, written language is a relatively recent cultural invention. It's only been around for 5,000 years. It is a product of cognition, a product of culture. So in preschool, children pick up words in speech and usually can recognize roughly 10,000 words by the time they enter literacy. Unlike speech, and we're all talking about this, reading doesn't usually come naturally. Kids don't magically figure out the code. As we heard yesterday, we need some balance of information about oral language and phonics and the code. Why? Because reading is this rather complex reorganization of the brain. Stan DeHaan refers to this, the fact that the brain is not wired to read and write, it is wired to talk and listen. So reading is an exercise in neuroplasticity, or what he calls neuronal recycling. You take a lot of systems or circuits or hubs that do lots of different work, like learning, perception, attention, memory, language, even higher order cognition. Remember, when a kid is in first grade, one of the things they need to do is sit still and inhibit certain responses, become focused. All of this is executive function, attention. Reading depends on all of this, but ultimately what it is the process of making connections between the visual representation, which is arbitrary, and the biology for language, which is written in the genes. So the cognitive challenge of learning to read in any language, shallow or deep, or alphabetic or non-alphabetic. When a child enters the school environment, as you've all been saying, they should know a lot of words in terms of phonology and semantics. So this part of the triangle, this is Mark Seidenberg, one of our colleagues and collaborators, this is his division of labor model. It's a model of how the brain, the cognitive system, is building a reading circuit. And so when a child comes into the classroom, they already know lots of words by phonological form and by meaning. Of course, what they don't know is anything about this rules of writing. So what the child needs to do is to create an integration such that, as Linnea taught us yesterday, in the early stages, they understand the principles by which graphemes represent phonemes. Or in Hangul Korean, the characters represent syllables. Or in alphasyllabic Hindi, the consonant vowel clusters represent phonetic information. This varies. Of course, what also varies is the grain size and the transparency from shallow to deep, from regular to irregular. But the child needs ultimately to build these connections. And they need to build them in such a way that what happens, you know, and I know, that a child who is a skilled reader is able to get from eye, when they see the word, the retina, to meaning. From a cognitive neuroscience perspective, we would estimate 250 to 300 milliseconds. So that's 1,000 milliseconds, for those of you not in these areas, is 1 second. So a skilled reader of any language is able to get from eye to meaning very, very fast. And of course, if you can't get from eye to meaning very fast, then, and it takes your entire soul to pull the word off the page, by the time you get to the end of the sentence, you will have forgotten the beginning. And so we often see in children with developmental dyslexia, with reading disability, specific reading disability, that the ability to pull words off the page is slow, labored, and especially in English, error-prone. But even in shallow orthographies like Finnish, it's slow and labored, and not automatic. And so the question in all of this is, what is the brain doing in order to build this triangle? So I ask you to keep this triangle in mind: orthography, phonology, semantics. Ultimately, at a cognitive level, you're creating a system with feed-forward and feedback connections that will allow you to get from eye to meaning bloody fast, okay? So how does the brain build these connections? And how does it depend on oral language? And so I'll be addressing 3 questions to keep in mind. 1, how does the brain solve this puzzle and build this fast mapping system that allows you to integrate orthography, phonology, and semantics? How does it do it when all goes well? Uno. Two, how does it differ in children who struggle with learning issues? And three, and most important, how does treatment or remediation modify those brain differences? So I will share information on all three of these topics. So first, let's consider the brain, and let's consider its biological specialization for speech. So I won't go into this in great detail, and I am oversimplifying this notion of Broca's and Wernicke's. For those who are deeply immersed in neuropsychology, I will tell you that every brain system interacts with every other system in a nonlinear, dynamic, self-organizing fashion, and so the brain is complex. And when you study it, it's complex. But just to give you a start into the biology of talking and listening, let's begin with this diagram. In the frontal areas, in the inferior frontal and supplementary motor, one region there referred to as Broca's area, if you damage this tissue, you will tend to have expressive difficulties and not necessarily receptive ones in oral language. So it clearly plays some role in speaking, in coordinating, and it also plays a role in syntax and lots of other grammatical processes. But it plays a role, it's an output, it's a motor system. In posterior superior temporal gyrus, in red, is an area called Wernicke's area. If you damage this tissue, you are more likely to have problems with receptive language and not necessarily with expressive. Now, that rarely happens. Everything is complex and interactive. But we know pretty well that along this left perisylvian brain, from the posterior temporal parietal to the inferior frontal, is a whole lot of tissue that is specialized for receptive and expressive language. And before you learn how to read, before you make that triangle I'm asking you to remember, you have information on the visual letters that goes where in the brain? Well, you know, it starts in the visual cortex. That's where we see the letters. And then somehow the job of becoming a reader is to create and find pathways that connect vision to language so that you can do what? So that you can get from I to meaning bloody fast. So that you can therefore put your attention to the syntax, to the grammar, to the pragmatics. Or, as Linnea was showing us, you can create a good memory system that allows you to put your attention beyond the word and to the higher order content. But somehow at the level of brain, it obviously depends on finding pathways that will connect vision and language. What I'm going to be sharing with you in the next few minutes is evidence that I find very compelling from our lab, that we can understand whether reading will be successful or not successful by the degree to which the brain is able to use natural language machinery to support reading. That's where I'm going next. We use many kinds of brain imaging. You'll see many of them presented today. A lot of the data I'm showing you in this first talk, in this overview talk, is from fMRI, functional magnetic resonance imaging. In an MR machine, the child, the subject, the person is in the scanner, and if they're using different parts of the brain to do different things like attention, memory, reading, language, there is a perfusion of oxygenated hemoglobin that takes place after the population of neurons is active. And that perfusion of oxygenated blood can be very precisely localized to within about 1 millimeter and can tell us where in the brain things are happening. We also use measures like EEG or MEG, which gives us beautiful information because it's measuring electrical activity or magnetic activity under the scalp, and gives us exquisite information on the timing of brain activity. A proper neurocognitive model, you won't be surprised, needs both time and space, where and when. And so we tend these days in everybody's lab to try to integrate modalities. But most of what I'm talking about today are where questions. Where in the brain is reading? How does it depend on speech? How does it differ in dyslexia? And what does treatment do to those brain differences? So let's think for a minute about what we know from brain imaging and from neuropsychology about the spoken language representation. Now I'm talking about whether you know how to read or you don't know how to read, which brain regions are going to be very involved in talking and listening and understanding, comprehending. In a sort of a well-characterized model by Greg Hickok and Dave Peppel, which has been revised over the years and which I quibble a lot with their lack of articulatory involvement. Nonetheless, it shows you that in the left hemisphere, shown here, there's a lot of populations or hubs that are involved in perceiving spoken words. The information comes into the auditory cortex here, and it passes either in a ventral direction to middle and to inferior temporal gyrus. Again, this is more for the neuroscience people in the room. For those not in neuroscience, I'm still going to tell you the big picture, but you don't need to remember the MTG and the ITG and the SMG and the IFG and on and on. But for those in the research, there is a more dorsal system that seems to be phonologically analytic for spoken words, and a more ventral toward the bottom system that is more involved in lexical semantics in the spoken mode. There's also evidence of right hemisphere involvement. Of course, speech is not entirely left hemisphere. It is left hemisphere dominant, though. So if you're not an infant, where laterality doesn't matter, and you're greater than 4 years old, if you damage left perisylvian tissue, you are much more likely to lose language than if you damage the right perisylvi region. But you should note in this model that the right hemisphere seems to play a role more in prosody, in slow processing of speech information. In any case, that's what speech kind of looks like in the brain. What about reading? Well, we've been studying where in the brain reading activates for quite some time in my lab and many other labs. I want to begin in this first presentation with a very simplified general cortical account that talks about 3 broad regions. By the way, what these regions are doing is building that triangle in cognition, orthography, phonology, semantics, becoming adequately bound and feed forward and feedback integrated. But at the level of brain, we see that there is critical tissue in parieto-temporal, up here, and in the inferior frontal that our work in other labs suggests is critically involved in emergent readers when they are learning to crack the code, when they are learning to do the things we heard about yesterday. That the intactness, the integrity of the parieto-temporal and frontal areas, these more dorsal extending regions, is critical in learning to bind orthography to phonology, to semantics, and morphology. We're not going to talk about morphology, but it's interesting, and it's interesting in the brain. When the child goes from being a beginning reader in any language in the 1st or 2nd grade, they're usually accurate, but not fast. You all know this. And as children go from being accurate to automatic, there is a developmental shift toward a region that you're going to hear about in other talks today called the visual word form area. You're the Dahan guy, right? No, yes, there you are. So visual word form area, I always say to Stan, is a misnomer on 3 levels, but it's a very important bit of tissue, for sure. And the visual word form area it lives in ventral, fusiform, occipitotemporal cortex. It's a population of neurons that is really good at fast pattern recognition. In the right hemisphere, you have somewhat homologous regions that are very good at face processing. But all of these pathways are very good at fast mapping. And so as a child goes from being accurate to being automatic, they're reading fast, there is a developmental shift from the learning circuit into a more heightened role for the fast mapping system. How do I know this? Well, there's lots of data in lots of languages. This was our original data. We had 174 children between the ages of 7 and 17, and half of them were struggling with reading, diagnosed with dyslexia, and half not. But the critical thing that we found, and this is something you have to listen carefully for a minute. When you're seeing these brain images, these horizontal images, these are called axial oblique images, the left hemisphere is on the right side. This is a radiologic convention. I'm on faculty in the School of Medicine at Yale in radiology, and I still get annoyed, why can't you just flip it? But they don't flip it. So this is the left hemisphere on the right side of my screen, on the right side of that brain image, remember. And here is tissue in the occipitotemporal junction that has very much been identified as the visual word form area. And what we found these many years ago is that we could predict how fluent your reading is by how much you activate this region. We had a beautiful relation, and very few other regions in the brain showed this relation. So we knew, and we also found that it was correlated both with age, because the kids were from 7 to 17, and fluency. Turns out what really matters for those of you who are in research, when we did multiple regression, it wasn't your fluency, it wasn't your age, it was your fluency. But of course, as kids become more automatic, as they mature, they develop this liking to this particular fast mapping system that DeHaan calls the visual word form area. Now, I don't have time to go into this today, but I will just share this with you briefly. If you're interested, for those of you in the research, in the neuroscience research, we did a large study a few years ago, 2013, with 70 or 80 children who were emerging into reading, emergent readers, beginning readers. And we did a variety of complex analyses, partial principal components and partial correlations. We identified predictors, including measures like phonologic awareness and the like, that we know if the child is doing better, they're more likely to do well in reading, and you'll see that in a minute. And then we mapped into this beginning reader brain, and we were able to identify those brain regions that were more active in children who were further along in becoming literate, particularly in their phonologic skills. And the reason I have this slide here is not to go into too much detail, but you need to understand that the brain is a cortical, subcortical, amazing machine. And learning doesn't just take place in the cortex. So we have specialized subcortical areas, the basal ganglia and the thalamus, that work in conjunction with the cerebellum and with cortical regions. And they are critical in how we go from being beginners to being professionals at whatever it is we're learning. And this role of the subcortical regions, in our study, it turns out that the pulvinar nucleus was a very strong predictor of readiness to read. Now, I won't go, this is a bit of an aside if you don't mind for the neuro people, but the pulvinar is interesting. As many of you know, the pulvinar is sensitive to selective attention in man and primate. So when you have a complex visual scene, like all your faces, but I have to find one and ignore the others, this subcortical element, this pulvinar nucleus, which in its ventral aspect is retinotopically organized and its dorsal aspect projects into parietotemporal language areas, is very much involved in what the beginning reader is doing. What are they doing? They're building this sensitive marker. This is what letters are. This is how letters relate to meaning. So it's very important that we consider the full learning machinery. And I want to mention one other thing. I talked about the parietotemporal and the occipitotemporal and the frontal areas that are part of learning and the subcortical areas. But as teachers know, when children learn, they need to be able to have self-regulation, executive function, and we found that all those regions are involved in predicting which children are further along in learning to read. Now I get to the important slide. So please come with me on this for a minute. This will help us get through the rest of my talk. This is an fMRI experiment with skilled adult readers of English. This is just a bunch of people who know how to read really well, and this is a 3D rendering of activation shown in these colors. What is that activation? It's very simple. In this experiment, the subject, the person, does one thing only. They have to get a word and decide if that word is a living thing, like cat, or a non-living thing, like chair. Very easy, but they have to go to meaning. What we manipulated is whether the word came to your ear, your natural language, or your eye, your reading system. So we are looking at, in this experiment, which brain regions are involved in reading, which ones involved in listening, and importantly, which ones are involved in both. So if you look at this slide and you know anything about the brain, you won't be surprised that in red, in the primary auditory cortex, are populations of neurons that respond uniquely to spoken words because they're auditory. And in the back of the head, if you can see this here, if I turn it around, in the visual cortex, in yellow, are populations of neurons that respond uniquely to print. If I don't see that, we're in trouble. If the primary visual and auditory cortex doesn't respond to print and speech, then I might as well go into another business. But what we see for many of those other regions that were part of the spoken language map, the Hickok and Peppel map, that are involved in talking and listening, here they're painted green, Wernicke's area, the angular gyrus, the inferior frontal gyrus. And what this means is that these hubs in a literate, skilled adult reader no longer care whether the information came to my eye or my ear. They have transformed and they have become what we call amodal. So these language perception and production systems have become changed by learning to read. Does this matter? Well, learning to read changes speech. Ana Luisa talked about this morning. When you learn to read, you're able, in the famous Castros-Caldas study, you found that illiterate versus literate adult Portuguese speakers, that literates are better at responding and shadowing pseudo words. Somehow, some non-reading behaviors of listening and speaking are impacted positively by learning to read. So we're interested in the bidirectionality. In another study by my colleague Manolo Carreras at the BCBL in Spain, they found that when you train and when you look at literate versus illiterate adults, the literate adults have greater gray matter volume in a number of those language areas. Learning to read has changed the machinery of speech. And in a beautiful study by Dahan and his colleagues, where they actually taught illiterate adults to read, and they became ex-illiterates, they found that in the auditory speech regions that I'm about to focus on, superior temporal gyrus, that having learned to read makes the tissue there, the system there, respond more vigorously to auditory speech. So we often think about how learning to read depends on speech, and it does. But it also changes speech, and we need to be sensitive to that relationship. Now I want to get to the really core content of our work on the other direction, on how reading depends on speech and changing the speech organization. So in this study, we're testing an old hypothesis, very much a Haskins hypothesis, that good reading is in some biologic sense parasitic on speech. So the integrity of the talking and listening machine, the Broca's and Wernicke's, the perisylvian regions, is going to be critical in the ability to create good literacy. And why this is going to matter, I'm not there yet, is because it stresses the importance of oral language and on making those connections that we've been talking about. But let me prove to you, if you don't mind, how important this integration, this transformation is. So we had a longitudinal study. We have nearly 100 children, this is a different study, that we tracked for 2 years, from age 7 to age 9. So when they were beginning readers to when they're in 3rd grade, and they should be good readers. And when they were beginning readers at age 6 and 7, roughly, we looked at the brain, these beautiful 6 and 7-year-old children. They came into the scanner and we measured their brain's response to spoken words and written words, okay? And we looked at this integration, this thing that I was talking about. And what we found is that in the left speech areas, the superior temporal gyrus, we found a very important interaction, one that should be intuitive, especially for reading specialists. In this part of the brain that is specialized for oral language, if you have good phonemic awareness or impaired phonologic awareness, phonemic awareness, it responds to spoken words the same because it's designed for spoken words. But what is so striking is in these 7-year-old children, if they're better able to do phonologic games, then this speech tissue is responding not just to speech, but to print. If they're having troubles with phonics and phonologic processing, then they have not made the connection into these perisylvian language areas. So we see that in this interaction, that children who are further along the reading readiness have started to transform these perisylvian regions and make them available for reading. Now, does that matter or is that just cool? Because in some sense, your grandmother could have predicted that finding if your grandmother were Isabel Liberman or Mrs. Orton of the Orton Society. Better reading means you can activate speech tissue. No surprise. But it turns out it really matters because we followed those children for 2 years. And in a paper we published in Psych Science with my, at that time, postdoc, Jonathan Preston, a brilliant speech and language pathologist, we followed those kids and we said, what was it in 1st grade, when I measured them, that best predicts 3rd grade reading? And 3rd grade reading is when you should be transferring from learning to read to reading to learn. So what is it in the brain of the 1st grader that best predicts how they'll read? Is it what the brain looks like when it listens to words? Is it what the brain looks like when it reads words? Or, to my hypothesis, is it whether it uses the same tissue to do both jobs? And it turns out we measure the overlap, the integration, and it turns out that the only thing that matters with all these statistical controls and regression is whether in these perisylvian areas you have created this amodal system. The best predictor of 3rd grade word reading scores is the degree of integration at age 7. The degree of integration at age 7 is between speech, language, and print. How could you not teach something that stresses these connections if this is in fact the best predictor of 3rd grade fluency? Is this true only in English? English is a crazy orthography, muy loco, right? Or if in Spanish, I don't know what you say in Portuguese. Crazy. But we've been looking at this in a number of ways. I have beautiful data on Chinese children we're just publishing now, showing that these things are the same in Chinese, a very different writing system. But I'll share this paper with you. We published this a few years ago with multiple laboratories in different countries in PNAS, and we looked at this transformation of perisylvian regions, Wernicke's and Broca's, with literacy. And these were all successful readers in Spanish, similar to Portuguese, it's transparency. English, not so much. Unvoweled Hebrew, really not so much, because there's no information on the vowels, for God's sake. And Mandarin Chinese with its irregular phonetic and semantic radical structure. And it didn't make a jot of difference in the brain. This transformation of perisylvian tissue that makes it so that it no longer cares whether the stimulus comes to eye or ear was in fact universal. And what we saw in Chinese children is that we could predict reading in 3rd grade, just like in English, from the integration in 1st grade, in a language that is very different. And it's all about speech. It's all about oral language. So cross-language similarities outweigh differences. One important thing for those of you who are more in the research side, just because you use the same tissue to do something doesn't mean that the something you're doing is identical. Computational differences might be present between Portuguese, English. I mean, there has to be some difference. But I would submit to you that if you're using the same circuit, and each population of neurons has its own tuning characteristics, its own character structure, then you're going to see more universality than less. This is, of course, an argument that Stan DeHaan has made eloquently, that really universality, the fact that at the level of brain, you need to transform all these circuits, you need to build the fast mapping, you need to transform speech cortex and make it literate as well, that all of these things are universal. And in fact, our work and his suggests that it is. But this also suggests that maybe there should be similarities in how important it is to teach the basic phonologic information that we heard about yesterday from Linnea, from Claudia, from others. I borrowed these slides from my buddy Manolo Carreras at the BCBL in Spain. These are under review. They're about to be published. But it was such beautiful data he presented in Argentina that I called him, I said, please give me those slides. Because he took a group of pre-literate children and he taught them to read in 6 months, sort of a sped up reading thing, one of 2 ways, whole or analytic. And he tested them on phonologic skills and semantic skills. And he started with the hypothesis that if you emphasize the analytic, the code, well, you'll be good at phonologic tasks, but maybe less good at semantic processing. And maybe the whole language approach would be better. Now, this is not a full-blown study along the lines of Hume and Caravalos and others that I think are very compelling in suggesting universality in what you need, even in shallow writing systems. But he found the following. Whether he was looking at your automaticity and speed at phonologic judgments or semantic judgments, it turns out that the analytic teaching strategy was associated with better gains, even in semantics, than an emphasis just on semantics. So you've heard a lot of this. I just want to add my voice. I think Catherine also yesterday said, look, we need a balance, but you need the basic elements, as Linnea told you. Okay, quickly. Our studies indicate, what have I been talking about for the last 40 minutes? The first question, really. 1, how do typically developing children in any language that we've looked at become fast and automatic readers? And there are a couple of things we now know. They need to reorganize and create a kind of a distributed circuit in left cortex. There are dorsal and ventral projections. The thalamus and the basal ganglia, we have other studies, are very important in learning to read. But ultimately, the signature of reading successfully seems to be this rather simple idea, I submit, that the ability to use the machinery of talking and listening for reading and writing is the key to successful reading outcomes. And we're not the only studies to show this, but the Preston et al. Data is gorgeous. I mean, we had a really big R-square, and we did all kinds of covariance. We even removed the same test at first grade, the autoregressor. And this brain integration of print and speech predicted above and beyond the behavioral test on what the reading scores would look like on the same test in third grade. So this brain data is giving us a value added in this regard. But it also suggests, wait a minute, if the ability to build a fluent reading brain depends on the ability to transform the mechanisms of articulation and reception, then shouldn't it be the case that early problems with articulation or speech perception should be predictive of reading problems? Not just the phonologic awareness that everybody fights about. Say the word cat without the first sound and you say at. That's a metacognitive. It's an important measure and it works. But shouldn't we see if what the brain needs to do to read is to kind of transform all these circuits? Shouldn't we see something that would suggest that early oral language in preschool will predict? So we have about 15, 10, good. So this is a study where those children that we were doing longitudinal tracking, when we were waiting for them to finish 2 years of development, while you do a longitudinal study, you wait around for kids to get older. We actually did a study where we had a history of the children, and we identified kids, and Anna Luisa will know this, who were late talkers, who had delayed onset of speech. Now, Heikki Luutinen and lots of other researchers have shown that language delays, expressive language delays in 1.5 to 2-year-old children are significantly predictive of reading problems later on. Not every poor reader was a late talker. Not every late talker is a poor reader, but there's a relation, one that's exacerbated by family history in the Finnish studies. So what we did is we had these children, we had beautiful brain data at age 7, and we had their histories. So the first thing we said is, do we replicate this finding that early expressive language problems are associated with poorer reading? And the answer was yes. These are the late talkers, these are the on-time talkers, and these are the precocious. This is word attack and letter word ID from the Woodcock-Johnson. These are pseudo-word and word reading tasks. Now remember, not every child who's a late talker is struggling with a diagnosis of dyslexia, of course, but they're reliably worse. So what do we see in the brain? Well, it's hard to see on this. I apologize. This did not come out quite the way I wanted. But if you look here in the thalamus, in the basal ganglia, and in the superior temporal gyrus, you'll see these little red dots, hard to see. What we found is that the children, even at age 7, who had been late talkers, showed anomalous processing in the cortical subcortical pathways, and this was correlated with their reading scores. So early oral language problems put reading at risk. I don't have time to do this. I wasn't sure if I'd fit it in, so I put the slides in. I'll just give you a very brief tease on this. We've published a number of studies in the last couple of years with colleagues in Holland where we look at something even more basic than the speech accuracy, the speech gestural complexity. And it's called, we use a task called the modified auditory feedback. I don't have time. I sort of watching my watch. I wish I had time. But it's a task where the child has to keep repeating a word over and over again. It's a boring task. And at some point, the information is coming back to their ear, but we transform the signal with the computer. And when we transform the signal, their brain gets a message. It's completely unconscious. Children don't know, nobody notices it. But the brain gets a message, hey, you're supposed to be saying shoe, but you're saying sue. It's like a little trick we play on the brain. It's harmless. And what we're doing, and the brain, and the child doesn't know, but what happens is when the receptive areas get an error, they change the way you're producing the speech. So it's a kind of a feed-forward, feedback dynamic. And it's really bizarrely simple and unconscious. It's a basic receptive expressive dynamic. And it turns out in 2 studies, it discriminates typical from dyslexic readers. So dyslexia has nominally nothing to do with modified auditory feedback or Broca's-Wernicke's dynamics, and yet we can discriminate that. And this last thing, which I don't have time, I wish I did, is really beautiful. This was just published this year, but it turns out, like the Portuguese illiterate-literate study, these are pre-literate kids versus literate kids. So they're all going to be literate, but some don't yet read. And it turns out the ones who can read 30 words or more on the Woodcock-Johnson are better at this receptive expressive speech task. So already learning to read is getting into that speech system and transforming it. Okay, quickly. Dyslexia. What do we know about question 2? I've already given you a lot of information, but it really boils down to something simple. If you're typically developing, you take a lot of left hemisphere specialized regions, you create a circuit with a word form fast mapping system, but the circuit, if it has integrity, should become such that it no longer cares whether you're reading or listening. It becomes amodal in some sense. And of course, good treatment and remediation is trying to strengthen those connections. What does dyslexia look like in the brain? Question 2. Well, no surprise, it looks different. It has to look different. Dyslexia, of course, is a continuous statistical definition. That it isn't a dichotomous. Good reading ranges from superior to average to impaired. And what that means is that there's a gradient dimensionality in reading. It's continuous, not discontinuous. And what that means is when you're looking to the brain to understand dyslexia, you're not looking for dichotomies. You're looking for gradient continuum. It's also, and many of you should know this, it is orthogonal. Dyslexia occurs whether is orthogonal to IQ. So you could be a genius and struggle with reading, you could be average and struggle with reading, and you could be well above average, below average and struggle with reading. Reading is not about intelligence. Disability is not about intelligence. It is about struggling with building these systems. And we know that for children in every language that's been studying, including Spanish and Portuguese, that children with dyslexia find it inordinately difficult to build the base of that triangle, for orthography, phonology to be bound. And of course, our work suggests to transform receptive, expressive language systems to become literate tissue. And so this lack of activation, what we see when we look at the brain of children with dyslexia is a failure to show coherent activity in posterior cortex. And in this classic study by Araldo Palazzo, comparing Italian, which like Portuguese is more transparent, or crazy English, or intermediate French, the signature of dyslexia, the failure to build these efficient circuits, these integrated circuits, is universal. And where goes function? So this is what dyslexia looks like. It looks different. There's a failure to show coherence in these posterior regions. We also see it at the level of structure. And I think others will be presenting things on this today as well. So where goes function, there goes structure. And where goes function and structure, there goes something called functional connectivity. So in 2000, there were 2 studies that came out, mine and one by NIH, that were the first to show that what is best helping us understand dyslexia is not the activation or failure to activate a given region, but a failure to have regions talking to one another. This is called functional connectivity. Others might cover this. The failure to create these kind of feed-forward and feedback systems in left posterior cortex. So here is a summary slide. If you want these slides, they're going to be available. When we look at the dyslexic brain, they look different, of course. We see a failure to show coherent activation, connectivity. We see gray and white matter differences. But all of that information is not an explanation. So you should be asking me a critical question. How does dyslexia look different in the brain? I've already told you. If you're typically developing, you create these integrated word form systems. If you're not, you don't. You tend to go to the right hemisphere, you tend to go to other parts of the brain. Is that an explanation of dyslexia? No. It's only a description. Why does the dyslexic child go here rather than there? That's a deeper neuroscience question. If I have a few minutes at the end, I'll show you how we address that with neurochemistry and genetics. If I don't, I'll just allude to it. But knowing how dyslexic differs does not explain it. It's not good enough. We need deeper neuroscience. We're after that. But guess what? There is something good about it. Because if I know that typical development is associated with this left hemisphere speech print binding, this fast mapping visual word form, then I have a target for treatment. If a child is struggling with reading and they deviate from these circuits, there's a big question. Does the right treatment for the right child at the right age help that child to normalize? Does treatment effectiveness, helping children, does it normalize these brain differences? Or do you end up just using compensatory circuits forever? So many of us have taken this on. As I mentioned, we've found that if you're typically developing, you get this visual word form area on the left. If you're dyslexic in the absence of treatment, the older you are, the more diffuse the brain activity. So the question is, what happens if we do an evidence-based, phonics-grounded, vocabulary fluency treatment developed by my wonderful colleague, Benita Blackman, who is the only person important in this, one of the early studies on treatment in the brain. We had a large number of kids. They flew 3 times to New Haven, to Yale. We measured them pre-intervention after 9 months of 5 days a week, practice. And so at 12 months they came back, and then 12 months later for a follow-up. And what we found is that on average, the treatment affected positively about 76% of the kids. That's pretty good. And so what we found is that it normalized the inferior frontal, the parietal, and the so-called visual word form area, this very important fast mapping system. We found that the treatment, but not the control groups, began to show the shift to the left hemisphere and a reduction in compensatory right hemisphere involvement. And that's good and exciting. But here's the problem. Well, first of all, lots of studies have shown this in lots of languages. So the brain is very plastic, and if you get kids at the right age with the right treatment, you begin to see some normalization in many of them. The problem is this, guys. What about kids who don't respond to treatment? So one of the promises of neuroimaging and neuroscience is this. Because it has exquisite sensitivity to individual differences, we are now using it to ask this question. Can I, looking at that child's brain, understand why some kids respond to treatment and others don't? And by understanding where their brain has fragile connections, can I tailor the treatment to strengthen those? In other words, the promise of educational neuroscience, with its exquisite sensitivity to individual differences in brain circuits, is what we're now looking at in our new NIH studies, which is, can we understand not just what treatment does on average, because we've already done that. On average, when treatment works, when we remediate dyslexia, we get a beautiful increase in the left hemisphere. But why does it work for some and not others? Who are these children? What do they need? And so this is a study we just completed with Robin Morris and Maureen Lovett and her beautiful treatment called Empower. 150 children, 80 hours of treatment. And here's what we found. On average, we replicated all our previous work. And after treatment, those left perisylvian areas start chugging away and doing a good job with reading. But we don't care this time about average. We care about individual differences. And here's what we're finding. That we can identify certain brain structures and functions that predict whether the child will respond to treatment or not. And once we know what these regions do, we can begin to rethink the treatment. What will that child get if I add a little more executive function, a little more of this? And this is, of course, the promissory note. To end quickly, this is for the imaging people. Sorry, I'm throwing this in. We have a separate NIH grant that I'm doing using the same treatment and 80 hours, but it's doing something we should have been doing a long time ago. And when we do treatment work around the world, we look at the brain pre and post, and we try to figure out what it is pre that predicts whether you're going to respond to treatment. It's great. But for those of you who've ever treated a child and taught a child who struggled with reading, you know that when you start treatment, they don't just start going up. They go, nothing happens, nothing happens, nothing happens. One day they come to class, bing, a light bulb goes off and they start to go up. I want to know what that light bulb is in the brain. But if I can't know it if I look only at the beginning and the end. So what we're doing now in my NIH studies is we're looking at frequent imaging during treatment, trying to understand which connections in attentional control and other systems are affecting these brain pathways. So the treatment work is going very much now into individual differences, trying to tailor the learning to the biology. We are not in a place where we should be bragging at how far we are. But all of this NIH work that I'm doing and other labs are starting to do is really suggestive of the power of cognitive neuroscience, which is to understand variation and not just the average. Let me end with just one last slide, if I might. As I said, when you know how dyslexia looks different in the brain, and I've already gone through that, you have a target for treatment, and that's awfully good. And then you can ask, why do some kids respond? What do other kids need? And all that good stuff. But why the dyslexic child struggles with creating coherent connectivity in the left hemisphere is not answered by fMRI alone. We need to dive into gene, brain, behavior analysis. We need to dive into the brain's chemistry. So please let me end with this. I won't be able to justify it. But we need to go beyond descriptive. Much of brain imaging today is correlational. If you're a better reader, you go here. If you're a worse reader, you go there, whatever. We need now to understand why certain genes, mixing in certain environments, produce this unstable lack of reorganization of speech areas. And in order to do that, we need to dive into animal models, computational modeling, multimodal imaging, and lots of things that many of you never have to do, but I have gray hair because I'm trying to do these. And so, for example, this is some studies we've done looking at a specific gene that is associated with cognition, the COMP gene. And this is a gene that has a specific allele, the Val-Met allele distinction that is associated with better or worse executive function, memory, and some other functions. We were able to link it to reading, but then we were able to look at the brain in children who had these different genetic variants. And what we could do is make a sensible three-way relation between risk genes, neurofunction, and reading. And so this is, of course, we're in kindergarten, even the best labs in the world are still in kindergarten when it comes to genetic imaging. We're doing a large study with multiple countries right now at Haskins where we're borrowing everybody's data around the world we can get that has structural MR and genetics. And we're trying to do what's called imaging genetics. And the last thing is become a highlight for my group and for some of my NIH grants with my colleague Fumiko Heft, H-O-E-F-T, who's become a real leader in this field. And here's what we did. We measured, while children are watching a movie, we measure using an MR machine, a magnetic resonance machine. We're able to, in a very large region of the brain, and 7 years ago, we can measure the chemistry of the brain. You're watching a movie, it's entirely safe, but we're able to quantify the major neurotransmitters, particularly GABA, the major inhibitory neurotransmitter, and glutamate, the major excitatory neurotransmitter. Now, for those of you who are not in the field, you're not going to remember all these details. I want to share what we found, though. We found that we could predict at age 7 whether a child was going to struggle with reading, and we could predict their reading scores 2 years later from whether they had too much of the excitatory or too much of the inhibitory neurotransmitter in certain brain regions. This opens a doorway. This is not for prime time. We're doing an entire grant now using multiple imaging and genetics and EEG and MRI and functional MR and behavior in order to understand what the neurochemistry of learning difficulties are about. If you look at this last slide, remember it. I'm going to probably drop dead of a heart attack in the next year because we're trying to do everything from the bottom up. Genetics, EEG, MRI, neurochemistry, behavior, learning, adaptation, etc. What science needs to do, and I'll stop here, what science needs to do is we need to bring many disciplines from genetics to neuroscience to linguistics to bear on mechanism. We still don't have causal models. This is the way we go after causal models. But we can be happy about what we've learned so far with neuroimaging, which is we can identify objective differences and exquisite sensitivity to individual differences, and we can explore things like what works, for whom, and why. Thank you. Okay, we have time. Do we have any?
Yes.
Where's my mic? Okay.
I just need to read it to the
Yeah.
translator. Eu vou ler a pergunta em português e vou traduzir, como eu já combinei também com os tradutores. Tá? Tem mais perguntas? Eu acho que I'll read it in English to you. Don't worry. So it doesn't get confusing. Tem algumas perguntas específicas aqui sobre perda auditiva e eu acho que também vocês podem depois direcionar diretamente para ele. There are some specific questions about hearing loss and I think they can talk to you. One question, and then I'll translate it as well, is what does Haskins do locally and worldwide to inform public policy, and where do you think academia is failing that process? A pergunta é o que o Haskins faz localmente e mundialmente para informar políticas públicas de educação e onde ele acha que a academia está falhando, digamos assim, fracassando nesse processo.
I have, while I answer this question, if we can put up my slide, because my last slides are about what we think we can do for policy. So it's the very end, toward the bottom. And this will make it easy for me to answer very quickly. Way toward the end. What we believe, first of all, I do a lot of work, as many people here, on international panels and advisory councils and things like this. So go back, Augusto, just maybe to where it starts to be black. That slide, yeah. So, okay, doesn't matter. So here's the thing. It is critical that we have 3 partners at this point. I've created something at Haskins, and we are already collaborating with a number of people here in Brazil, called the Haskins Global Literacy Hub. It is a multidisciplinary group. We're collecting data right now in Argentina and Mexico. We're about to begin data collection in Brazil with Augusto Buchweitz and his colleagues, and we're also doing work in Chile. No, I'm sorry, Peru. And next year in India, we're starting at this point. What is critical for us as researchers is to do 2 things. One, we need to actually work with teachers and educators in a respectful translational partnership. For neuroscience to deliver on education, educators need to learn from neuroscientists, but neuroscientists need to learn from educators, people who understand how to teach and understand what the questions are. So we've created these new models where we work in schools. We bring some of the neuroimaging, the EEG and some others, into the classroom. But what we do is we also work to identify best practice with educators. And then finally, having created these kinds of models, and right now we're doing it must be 1,200 children in 2 large schools in the US that we have the EEG lab in the school. And we're able to work day by day to understand these children more richly. But we're also learning from the teachers. So I think it's critical that educators and researchers listen to one another. And of course, the third part of the triangle is obvious. If they listen to one another, they have more power and more force to deal with policy and with politics. At the end of the day, the only hope we have to transcend politics is science, as this conference has been saying from day one. But science will get better if we are willing to work in truly transdisciplinary ways. So what we're doing is we're trying to take that very seriously. In fact, I'll just quickly mention, these are some of the people who are advising this project. People that are leading in computational modeling, Mark Seidenberg, Elena Grigorenko, Ludo Verhoeven, Heikki Luutinen, Linda Mays, who directs the Yale Child Study Center and does early brain development, Richard Aslan, who studies how babies learn language. We're all working together, and we're working with schools and with educators to learn from each other. I think that if we do this right, and frankly, what I see going on in this work here in Brazil, and Hanan and all the members of the reading panel, the National Literacy Panel, is that first step to bring the partners together. I think we can have an impact. I think science can have an outsized power if we are able to make sure that we're getting accurate information and that we're getting the best practices. So right now, I think the best solution to bring this to scale is interdisciplinary partnerships. And that was